I am delighted to introduce Professor Joe Jez. He's a Spencer T. Olin Professor of Biology and a Howard Hughes Medical Institute Professor at Washington University in St. Louis. Joe was an undergrad at Penn State in 1992, completed his PhD at the University of Pennsylvania under Trevor Penning in 1998, and was an NIH postdoc fellow at the Salk Institute between 1998 and 2001 in the lab of Joe Noel. After working at Cosin Biosciences from 2001 to 2002, he started his group at the Danforth Plant Science Center in 2002 and then moved to Washington University in 2008. He has just completed his 5-year term as the department chair. He has received the Presidential Early Career Award for Scientists and Engineers, Phytochemical Society Niche Investigator Award, Fulbright Senior Specialist Award, American Association for the Advancements of Science Fellows, and the Arts and Sciences David Hadas Teaching Award. Research in the Jez Lab uses a combination of structural biology, protein chemistry, and plant science to understand the biochemical pathways in plants and microbes with the aim of engineering these systems to address agricultural and environmental problems. We're very looking forward to hearing what you have to say today, Joe.
So thanks everyone for being here and for the introduction. So I will upfront admit I am not an AI or data scientist, right? I am a practitioner of X-ray crystallography and biochemistry. And I will say this though, it's been amazing to see what has happened and what is still to come. And I actually want to thank you have a beautiful setup for some of the things that I'm going to talk about. I'm not going to dive in too far. Next slide, please. So like I said, I'm a crystallographer, a structural biologist. I tend to think about proteins from a very structural mechanistic regulation component. But ultimately, I also want to know what those proteins are doing and how we change them impacts metabolism, cellular processes, and ultimately organism fitness, right? So we try to think about crystallography as a hypothesis generator that lets us move through different scales. Next slide. So all of you in this room are actually, well, if you're doing bench work, you are a structural biologist in some way, shape, or form, right? And think about the birth of molecular biology. Watson and Crick and Rosalind Franklin and Maurice Wilkins doing the first diffraction, fiber diffractions of DNA, looking at the structures of what a helix looks like. It's really about thinking about biological and chemical information in 3 dimensions and thinking about molecules and what they do. I always use this slide when I teach. I point out that in 1962, right, there was a Nobel Prize in medicine for the structure of DNA, but there was also a Nobel Prize in chemistry for the structures of myoglobin and hemoglobin, the first proteins. And before we jump to the 21st century, I want to talk a bit about the 20th. Next slide, please. Because it sets up a lot of the pieces of what's really happened in structural biology in the last 20 years or so. So go back in time, almost 100 years ago was when the first proteins crystals had diffraction patterns. Took another 30 years for Brutz and Kendry to get the phasing issue. Back in the mid 60s, if you had a crystal structure, it was a suit and it was a tux and tops type of event, right? You got to have a lot of PR around that. Now it's common day. 1970 was the birth of the protein data bank. And frankly, for many years, it was utterly unused, right? Because it was so slow to get crystal structures. By the 80s, there might have been a dozen proteins in that data bank. Think back to the technology we all have in our pockets right now, the cell phones, the visual technology on the cell phone. Back then, you needed a room just to visualize something on a graphics terminal, right? All of these pieces begin to accumulate, high throughput screening, accessible graphics. And in 2000, the Protein Structure Initiative was launched. And the PSI really, was a huge NIH funding effort, a lot of big initiatives across the country, across the world, actually. And it really pulled all these threads together. And it had an incredible impact. At the time, many of us as crystallographers would poo-poo it as a boondoggle. Looking back, yeah, maybe some of the science that came out of it wasn't what would have come out of individual labs, but the infrastructure it built, the technologies it created enable what is to come. So next slide, please. To give you an idea of the scope of this, this is the number of depositions annually from about 1970 to 2025. And notice the green line. That's the 20th century, 21st century split. What we thought we had 20 years ago was incredible. Look at what's happened since then, right? Last Friday, I looked at the PDB. There's almost 218,000 structures deposited. Probably 20 to 30,000 unique folds. And many of these are copies of things, but that is an incredible amount of information that's now getting tapped. I'll point out, X-ray crystallography remains the tool because it is really the tool that gives you the atomic resolution, the details of how things are bound and what changes between apo and holo or different forms. CryoEM is taking off, right? This is enabling larger scale ensembles to be looked at, assemblies, complexes, and NMR is still there. It is a much more difficult tool to use to do de novo structure, but NMR also gets you conformational dynamic information and other pieces that aren't accessible. And that's still a technology that's lagging. Now, the other piece of this is you'll notice on the other side of that is the plus 1,068,577 computed structural models. Absorb that for a fact. It's taken about 40 to 50 years to get 200,000 experimentally determined structures, and almost overnight, with some of the tools that Mohammed mentioned, there's 1 million data structures out there. That is phenomenal, and that is part of this acceleration that's going on. Next slide, please. Right, so crystallography, there's 5 basic steps. I just point this out because if I was to have another 40 minutes to talk, there's all sorts of innovations in the methods and approaches here. The robotics that go into growing crystals, right? The molecular biology tools for purifying proteins, the synchrotron sources and what you're able to do with beamlines now is amazing. And it accelerates it. In the time I've been talking, I could have had 80 data sets collected. And the limitation is not the data collection or the robot. Well, part of it's the robot going back and forth, but it's also the read-write times on the computer files as you're moving gigabyte information across from the machine to the servers, right? So all these technological pieces are phenomenally built into what we do. Next slide. I just want to drop in for CryoEM, right? People have been doing cryo-EM structures of virus particles going back to the 80s and 90s. It's not a new technology, but it's actually the computational power and the imaging devices that we now use that have really brought this forward to a very user-friendly, relatively user-friendly approach. But it's also, you can already see it in the data, it's accelerating how we're getting new structures and building into these data pieces. Next slide. Now, the other part that probably I need to just point out, sequence explosion. And keep this in mind, right? Because a lot of the AI-driven folding systems rely on this, the 30-some million sequences that are in the databases. And as people move into evolutionary niches and broaden out the scope of what organisms we're sequencing, microbes, plants, archaea, fungi, and other higher order eukaryotes, right? We're getting more and more information. This is only going to improve those models that we're starting to see. Next slide. So sometimes when people say AI, you get how, right? This is most people's first experience. It's not a good one. But imagine if you could say, Siri, design me a biocatalyst that gives me a carbon neutral synthesis of a compound that my organic chemistry friend can't do. Right? That's almost the dream of what you want to be able to achieve with the technologies, and try not to think about how. Next slide. So Mohammed did a really great deep dive into AlphaFold, right? And I also want to put up a paper that I don't know if it's come out yet, but it's been on the science website about Rosetta Fold all atom, right? The ability to use deep neural network applications, AI-driven, machine learning, whatever you want to call it, to mine that data that we've spent the last 40 years doing, right? Generating the sequences, generating the structures, and being able to map them. Now, I'm going to give you a curmudgeon-y crystallography perspective. I don't necessarily agree with it. AlphaFold doesn't do anything useful. it's not doing anything with first principles, right? I can give it a sequence and it's not folding it de novo based on, what you would imagine going from unfolded sequence to intermediates to molten globules all the way through. It's basically back engineering based on sequences and known information and then fudging the rest of it. But now imagine you're working in the 20 to 30% of the genome that has no relationship to any other sequence. And that can be in a plant, a microbe, pick your organism. This offers you a way to bootstrap to something to say, hey, this looks like its shape, its sequence doesn't look like a kinase, but it sort of folds like a kinase. It lets you think about a hypothesis and starting point for how you might want to test things. And I'll come back to Rosetta Fold in a moment. Next slide, or one click. Now, the thing to keep in mind about AlphaFold is, yeah, it predicted a lot of structures, but just look at the very first page of those computer predicted structures in the PDB. Look at the images there, right? You'll notice there's 2 or 3 that actually look like a folded protein. That's pretty good. AlphaFold does really well when you have that data, right? Mohammed talked about this. But things that begin to fall apart where you just have weird random sequences or loops kind of out in nowhere, if there's no thing to ground it in, known information, and it can try to start to fold, but at some point it doesn't do that. So I've seen people present things as, oh, I have an AlphaFold model of whatever protein I'm working on, and it looks a little bit like the spaghetti string where it just goes off into nowhere. And it's like, well, okay, the core piece is probably right, but you still don't know how this is interacting. And also AlphaFold, has the problem of getting that accuracy of atoms within the structure. Think about what makes glyphosate resistance, the trait, really useful. It's a single methyl group difference in the protein, right? We're talking structural biology being a game of angstroms. If you really want to be able to engineer something really accurately, a ligand interaction, a protein interaction, a new variant, you have to be able to get to that resolution. Now, AlphaFold is a fantastic first step to that, and the models keep improving, which is phenomenal. But just remember, you get what just treat it as another tool. Next slide. So there's a paper that came out, I think last year, where it's sort of, if you look at the author list, it's a bunch of the old school crystallography people, and they're right. It's an incredible hypothesis generator, but their argument is it doesn't replace structure determination. Flip the argument around. I don't really need to know what the atoms are. I just need to know what the thing looks like so I can start designing cell biology experiments or domain swaps or whatever it is you're going to be. AlphaFold is really powerful in that respect. Now, it is limited in scope, right? And I say this, yeah, there's a million structures, but they're all proteins, right? RNA still remains really difficult. How do you predict those components? How do you think about assembling individual proteins, as complexes? And you're starting to chip away at that. But I look at pathways and go, I want to be able to think about how I can combine 5 different proteins that may come from different sources to do different chemistry, or how do I optimize a pathway, right? I can have all this upfront information, but I still can't quite do all of those pieces. My question about the conformation and dynamics is really something else, right? As a crystallographer, we still don't get conformational information. Like, we might get a snapshot of one form, another, and another, and we try to, hand wave how we morph between them, but we still don't really understand, how we drive those conformational changes. Think about designing an inhibitor, right? Cytochrome P450s are a great example, right? They kill drug trials. They kill a lot of different experiments because they're sloppy enzymes. Their active sites are actually very mobile and can morph, right? And it's actually that mobility that we can't model, that we can't really accurately visualize, that really becomes the crux of how they function. And that's one of the open horizons. But ultimately, it is an experimental accelerator. I can use an AlphaFold or Rosetta Fold model and have a new CryoEM structure and use those to fit into that information that I may not have to wait for a crystallographer to build that piece or do refinements that actually get me there. So it's still a powerful tool. Next slide, please. I think one of the things that's ahead is the molecular design piece, right? Rosetta Fold has actually been really good. And it's actually I have no association with this company, Arzetta. I just know about them. It's a spin out of David Baker's, but it really taps into that design, build, test cycle of being able to use these conformational tools like Rosetta to get you out of an energy minimum, right? A lot of us approach, I want to redesign the binding site of my glycosyl transferase to recognize a different sugar pattern or whatever. And we all fall into what's in the immediate area. But think about directed evolution experiments that routinely show mutations that are nowhere near the active site can actually influence what the functionality of a protein is, right? And that's usually through conformational components, different propagations of folding. Rosetta Fold can help accelerate that, pop you out of those local minimums, and then start to think about, okay, maybe I should test this. Now you can synthesize DNA pretty cheaply, very quickly. You have all the tools to be able to run through that cycle. Next slide, please. I kind of want to just put these two up. One is actually a paper that had nothing to do with AI, but high throughput fragment-based crystallography. If I can have a robot that can crystallize 5,000 crystals of my interesting protein, and I have a library of 5,000 compounds, I can now spend about a week and snapshot every single one of those crystals with potentially a ligand bound. Pull out that data and use the fragments to then start to think about building a new drug target or lead molecule, right? And you also start to see different, more targeted uses of deep learning or AI-driven methods to start to build ligand design. Now, it's still very much knob and hole, right? But you're starting to see people approach this, on their particular problems, building up to that much larger scale. Next slide, please. So I borrowed this from actually a current opinions paper, which I thought was really useful to think about, right? And it's really busy, so I want to try to walk you through it briefly. If you think about it, on the one side is the scales. Like, what do we know about behavior of atoms, the quantum level, to protein complexes, to intracellular, to the whole cell? In blue boxes are kind of, what would be called integrated biology. We can do CryoEM, NMR, we can look at all those pieces and think about a native structure and new states, conformational changes, mutations, et cetera, at certain levels. But then we also have other tools for the cellular component and the cell biology and the whole organism or whole cell piece. We have now a lot of the tools for thinking about theoretical, generating those models, right? Thinking about Rosetta Fold, AlphaFold for different landscapes and for different tools. We also have the ability to do molecular dynamic simulations, right? As people have started to think about how to do MD simulations from, really intensive to using a little bit more of a gradient to kind of enable longer term simulations, we can start to tap into those conformational models and things. And then ultimately virtual screening, right, is now also starting to bleed into a little bit of those theoretical pieces to combine. What I want to point out is we still don't have the tools to integrate across scales really well. We can think about individual proteins and complexes. We can think about cells. We can think about, what's going on with an organelle. But we still don't really have a good way of linking between those different components. And ultimately, right, we can do a really fine job on targeting a particular protein, but ultimately, how do we connect that into the whole systems? And that was sort of my question about, thinking about conformations, but even longer term, right? By 2120, when you guys we're all gone and they're having a Barris Fellow in the symposium, are they going to be talking about designer cells, right? How do we use the genetic information and the genome information and the time resolved components of that to start designing cellular factories in real time, using a lot of the data that we have at this point? So it's kind of fun to speculate. Like I said, Siri, design me a cell that does X, is kind of the long-term goal. Next slide, please. And so I would argue that in many ways, right, if you think about what we do from the genetic to the molecular to the cellular to the organism back to the field and the environment, you're seeing the impact of AI in all of these different pieces, right? And it's really cool because we have the tools to move through all of these scales. And AI is working individually in each of them. But I think long term, this is going to be another one of those challenges of how to use AI as an accelerator, not just within each of the boxes, but across all those boxes. And that's a problem way beyond my scope. But it's kind of neat to see some of the things that have been talked about at the symposium yesterday and today to really start thinking about that. So with that, I'll stop and answer questions.